Our final talk before the break is by Dr. Alessandra Vigilante from King's College London. Dr. Vigilante actually is, I think, one of our first fellows from King's who joined us relatively recently. She's a bioscientist, and she operates in that kind of interesting interdisciplinary realm between data science and biosciences. And actually, we met in person for the first time at the launch of Ali's hub for Applied Bioinformatics, which is set up at King's. And I'm sure she'll talk about that today. So thank you.
Thank you so much. Thank you so much for inviting me. It's a pleasure for me to be here and to hear this inspiring stories and presentations. So a bit about me. I'm a senior lecturer in bioinformatics, and I work at King's College London, based at the Christ Campus, London Bridge. I have a very nice office with a very nice view on the 20th floor. So if you come and visit me, you can see London. And so I lead a group, Drive, Lab, just computational. I have a background in biology. I'm a biologist, but I became a bioinformatician during my PhD, and I decided then growing up also through a postdoc and then group leader to stick just with bioinformatics and then collaborate with scientists and wet lab scientists. As Daniel was saying, I recently established the Hub for Applied Bioinformatics, and I also recently established Applied Bioinformatics MSc. As I was saying, I really believe in interdisciplinary efforts. There are so many scientists that need bioinformatics. Now, basically, it's very fair to say that there is no big research project without involving some sorts of bioinformatics and data analysis. And so people like me, with my background, they understand the biology, but I also understand the coding and the programming, were kind of really needed. In fact, one of the things that my mentor told me when I joined King's as group leader, she was like, one thing that you have to learn is how to say no. And I quickly understood why. So my group, the expertise of my group are mostly in genomics, transcriptomics, and data integration, and understanding the link between genotype and phenotype. Now, I know that here we all have the completely different backgrounds, so I'm not going to go into any big details of my research, but I just thought to kind of show what I mean by data integration, and now how I think that by using these data integration efforts, we can actually understand the big picture and hopefully answering this type of questions of development and innovation. So bioinformatics is very interdisciplinary. My colleagues are chemists, physicists, biologists like me, data engineering, and as I was saying, the volume of data is huge. Just yesterday, I was actually in Birmingham, invited by the UKRI for a workshop about data making the data findable, accessible, reusable, or interoperable. In particular, it was a workshop about interoperability. And it was so fascinating because also there, we were scientists, we were people from completely different backgrounds, everybody applying to UKRI funding. And, even if data are completely different, these are life science, in my case, data, but we were talking about engineering data and all different types of data. We all end up with the same problem of making data interoperable and stuff like that. So it is very important to think that, we all face similar problems when we work with data. Now, data in bioinformatics come from there are very different types of data. I deal with genomic data, with what the our DNA tell us, and transcriptomic, so RNA, and but we all, in different projects, I might deal with environmental data, diseases. I was working with electronic health records that are a very powerful tool that we have here in the UK, drugs, nutrition, lifestyle, and I think that just by linking the information from our genome and our transcriptome to our phenotype that might be a disease, it might be exactly the environment, the aging, we can actually answer bigger questions on personalized medicine, accelerating drug discovery, and public health innovation. So as I said, I'm going to go through 3 example projects of what I mean by data integration and which type of data I deal with. The first one is where I try to uncover hidden patterns, and we talk about, it's a UKRI MRC program grant funded, and it's in collaboration with a guru of HIV, and it's trying to understand, to find how the HIV works in its infection. So we started with cell lines that are taken from biopsies, skin biopsies of patients. We reprogram them in induced pluripotent stem cells. So basically the cells that then can become anything, skin cell, muscle cell, nervous cell. And we only took 150 cell lines. This is from an IPSI database initiative. That's what we do. We take these cell lines and we do a few assays where we test the HIV infectivity and replication. And we measure how much HIV infects and replicates these cell lines. And some of these cell lines come from the same donor, so they share the genetics. Some other have completely different donor. And what we see is that some cell lines are very easy to infect, so are very susceptible to HIV. Some are very resistant. Some are, once the HIV infects this cell, in some cell lines, it replicates very easily. In some other cell lines, it can't really replicate very easily. So this is our phenotype in this case. And basically what we're asking is why? Why is that? Because we know that actually, if a cohort of people is infected by HIV, there is a different outcome in terms of how quickly it develops into AIDS and how quickly the disease goes. So are we talking about something similar? Does this have a genetics based? And one thing is that these cell lines from this initiative have all sorts of data. So we have RNA expression. So we basically took these cell lines and we said, okay, what are the genes that are differentially expressed between these 2 phenotypes? And these are some examples. So for example, here we have on the x-axis the same cell line that I think is a resistant cell line, and on the y-axis 2 different susceptible cell lines. And what we were expecting, basically each dot is a gene, they all have the same expression, but then you have those outliers, and those outliers might be the ones that are associated with this different phenotype. And when these outliers always come up when you compare different phenotypes, they have a good chances of being involved. But we also have the DNA sequence, and so we looked at all the SNPs that are, so single nucleotide variation. In a certain gene, you were supposed to have one base, instead you have another one. And so we are looking at all the genes that are variations in common between the phenotypes. So hopefully to find something that can explain these different phenotypes. We even go further than that, and we took some blood samples from the multicenter cohort study in the US. They were basically collecting blood samples from patients back in the 80s, right? And they were freezing them. But what they have the clinical of these people. If the AIDS was developing very fast or was developing slowly. So what we did, we took these blood samples, it wasn't easy. We made induced pluripotent stem cells. We measured the same infectivity and replication as we did with our stem cells. And basically what we're trying to do is, if we can find the same gene, and we did genomics and transcriptomics, if we can find the same genes, and if we can go to the clinic as well. So this is one example. Another example, completely different, but still regenerative medicine. This is a paper that was published into Nature with my colleague of my department, Professor Francesca Spagnoli. And there the question was completely different. It's more about development. And it's something, liver and pancreas are completely different organs, right? And they have one fundamental difference. If you cut a piece of liver, it grows back. If the pancreas stops working, that's it, right? And they come from the same progenitor cell. Just at a certain point in the development of the cell, there is a split, and the cell decides to be a liver cell, and another cell decides to develop into pancreatic cell. So what we are trying to understand is really the mechanism between all these different steps to see if we can find some key gene that actually is important in this differentiation, and in particular in this, the fact that the liver has this potential to regenerate. So this is a mouse embryo, and what we did, we took cells at different time points, and we did a single cell RNA-seq experiment. But basically, you have the cell, and you measure the expression of each mRNA in each cell type. And what we found, and that's why it was published in Nature, because it was very cool. Here you have the different time points and the different cells colored based on their their population. And we see that in green, green, yes, we have the pancreatic cells. In red, we have the liver cells. And we could find two liver cells, two types of liver cells. You see this clustering, and they were clustered separately. We're like, why are they clustered separately if it's liver? They had the fundamental markers of a liver population. But even if they had the fundamental markers of liver populations, they were different. So we obviously studied them, but most importantly, we saw these 4 cells. You might say 4 cells, what's 4 cells? But we were like, why there are these 4 pancreatic cells that actually are clustering together with the liver cells? What are these cells? So we took other data sets already published, and that's why the importance of the data that have to be findable, accessible, reusable. You don't want to do an experiment from scratch when someone else did it for other purposes. And we kind of integrated with our experiment, and we found those cells even there, in between liver cell and pancreatic cells. I didn't put the image here. But basically, we discovered this new population is actually a transition population. And now we just submitted actually another paper on this, finger crossed, that gets published, because we are obviously studying these cells more extensively. So you saw, I just gave you 2 examples, completely different, but it's the use of the same type of data, basically, to answer 2 different fundamental questions. This is another completely different example. This is another project. It's called Mind and Skin, in collaboration with Professor Carsten Flore. He's a dermatologist, and he's an expert in eczema, atopic dermatitis, in particular among kids. And when you have atopic dermatitis, you have chronic itching. If you have chronic itching, obviously you have skin inflammation because you scratch all the time. And this means that you might have sleep disturbance. You don't sleep well. So this means that you're not rested, and so you might have hyperactivity. In fact, some of the kids with severe eczema, they are compared with kids with ADHD, even if they don't really have ADHD. They might have breathing difficulties, and they might have some impaired cognition problems when the eczema is very severe. So in this big study that is funded by a medical research foundation, we are still taking, actually, it's still ongoing, a cohort of 90 children, 30 with severe eczema, and they're taking basically a systemic tablet treatment, 30 with milder eczema, they only take cream and ointment, and 30 control kids. And we take so many different types of data. So in this case, we have sleep measurements. So we go to the house of these people, and we put a sensor on the mattress, a camera that actually takes a video of them while they sleep for a few nights. For 10 nights in a row, they have an actigraphy watch that measures the severity of the scratching, and headband and a cap, an electric that looks at how. So this is the first time I'm working with sleep data, and it's very fascinating. And then we do an MRI to look at brain region interconnection and brain inflammation. They do cognitive tests, all kinds of different cognitive tests. we take blood tests for inflammation and to look at circadian rhythm, and skin tests to look at skin inflammation. And then we have to take all this data. Each set of data is very difficult to analyze already, because obviously there is lots of noise and so on. And then we analyze each piece of data separately, and then we will try to integrate everything with machine learning approaches to kind of like answer questions like, how does chronic skin inflammation affect the brain? Why do some eczema patients experience more severe mental health issues than others? Can we predict which patients are at higher risk of neuropsychiatric effects based on their inflammatory profile? And can we personalize treatment strategy, basically, based on this multi-layered information? So I hope that with these 3 examples, I showed you what I mean by data integration and imaging, and the fact that I deal with really all sorts, we deal with all sorts of different types of data. So obviously what happened is that while I was leading my research group, I was also training a lot of PhD students and postdoc, because now everybody wants to learn bioinformatics because they understand that this is the future. A bit like now everybody wants to do AI just because it's cool. So basically, I was hearing a lot of like complaints from my colleague and PhD students and postdoc, where there wasn't really a community of bioinformaticians. They didn't know who to talk to. They felt very isolated, and so I basically asked my dean, I'm from the Faculty of Life Science and Medicine. I wanted to do something to improve that. I wanted to do something to put people together and that to boost grant collaboration and so publication and upskilling and so on. And actually that was the best email I've ever sent, because the day after he came to me and said, with an empty business plan, and said, what do you want to do? So that was very successful. So I basically came up with the idea of the Hub for Applied Bioinformatics that was officially launched in December 2024. And the main idea of the hub is, first of all, we wanted to map the bioinformatics and in the faculty and promote collaborations. So I kind of like, I really, and now I basically through surveys, I know who does what. I divided the group in kind of like who does just bioinformatics, who has a wet lab and does bioinformatics, who only needs bioinformatics but has nothing to do with it. I hired a team of senior bioinformaticians that support key research projects and grant applications, because sometimes some of my colleagues, they write a grant, they know that eventually they have to, one of the outcomes is going to be bioinformatics data. They don't have the expertise in-house, and it's not enough really to hire a postdoc, but if they don't account for bioinformatics, they're not going to get the grant, because the reviewer said, who is going to analyze this data? So basically what happened is that my senior bioinformaticians are costed in their grants with a different percentage of FTE based on how much work they have to do. 10%, 20%, 25%. And this has been super successful. We have been costed already in 25 grants, like something like that. We create new expertise and promote knowledge sharing with a training program. And also, we have a dedicated space where we have a hot desk, and it's a seminar in a seminar room where we host seminars and engage in activities. So, as I was saying, when people use it, when they write grants, that's the other thing. Sometimes they write grants and they don't have the expertise to write the grants, the data management plan. Like yesterday at UK, right, we were talking about, data, right, and how to make it interoperable and so on. UKRI grants, the reviewer look for something like that. What happens to the data? What do you do with the data? How you manage the data? And people don't know what to write these type of things in the grants, because they know the experimental side. So we help on that. But we also help if there are existing projects and people have funds, and so we work it out as activity. As I was saying, we have on training, and what I think is very successful here is that we have a combination of online training and in-person training. And even for the online training, I really believe in online training, because people doesn't have always the time to come and stick with us for 3 consecutive days. Also, people have completely different knowledge background, so it's too fast for someone, it's too slow for someone else. So we offer the online training, but then we have in-person follow-ups of those trainings. So you come and for 3 hours and we do more advanced stuff. But we also have in-person workshops that we do, like we're doing some for Nextflow, for example, GitHub, version control, high performance computing, and yeah, we open it also to external, not just to King's College students. These are the online training that we have. This is a platform that I got with, I created the UK Right funding. It's called Innovation Scholar UK, and we really teach from basic fundamental coding skills and all the all around basically towards basically being able to analyze next generation sequencing techniques that are all like the omics, the big data. And basically you go on our website, then we do data club, research seminar series to connect with people. We have hot desks. So my idea is that people come to the engagement activities, come to the training and say, oh, you do something similar. Why don't we work together, even if we come from different groups, in different departments, in different campuses, and let's book a desk. So you go on our website, you can book the desk and you can work with us. I am planning a series on AI and machine learning because I'm tired of hearing, oh, but AI, machine learning, people don't understand that. We do machine learning already. Like, it's not new. Like, machine learning is part of what we do already. So I'm trying to teach people that. That's my mission. We are getting very big in spatial biology. In fact, we're going to hire a person just on spatial transcriptomics, in case you know someone I'm going to advertise soon. And basically, the main aim is equipping minds and enhancing methods. So I'm trying to actually get grants to expand this outside King's College London. This is the team. These are my two lovely senior bioinformaticians and my admin. And we are very social, so you can find us on social networks. And that's it.